HCI, human-computer interaction. Now we're moving towards a combination of behavior and computation and beautiful stuff in the brain. Our next speaker, we switched around, okay. Our next speaker is Jeremiah Palmerston, mapping of self-motion information in the posterior parietal cortex of human behaving status.
Hello. I'm interested in spatial navigation because it's an important function of cognition for many species in the animal kingdom. Prey species need to avoid predators. Foragers need to find abundant sources of food, and sometimes humans need to find a midnight snack. And so we know that in the wild, animals create paths that they use often, and humans build roads. And so my project is specifically focused on familiar route navigation. So the brain collects information in what's called an egocentric reference frame. We have different sensory apparatus that operate egocentrically. That is, they can only communicate information relative to themselves. So speaking in an egocentric point of view, I can describe the position of my computer as being in front of me at about an arm's length away. But I can also describe the position of my computer in an abstract or allocentric reference frame without referring to myself. And in that way, I would say that my computer is in front of the audience in room 3 of the Cognitive Science Building on the campus of UCSD, and so on. And I can do that without referring to myself. So the question is, how does the brain create abstract or allocentric reference frames from only egocentric information? So past studies on spatial navigation have showed us what brain areas are important for this task. And so we have a picture here of a rat's brain. And I've highlighted certain areas that are very important for spatial navigation and the construction of abstract reference frames. And these arrows represent the way that information flows through the system. So this lateral posterior part of the thalamus is where sensory information comes in. Visual information and vestibular information are two very important types of information for spatial navigation. Vestibular information you may not be very familiar with, but it's the information that tells us how our head is oriented and how we're moving in 3 dimensions. So this sensory information comes in from the thalamus, and it's projected into the entorhinal cortex, which moves the information into this curly Q looking thing called the hippocampus. The hippocampus sends the information to the retrosplenial cortex, which then transmits the information to the parietal cortex. Now without each one of these very important brain regions, then an animal would be completely lost and unable to find its way. So with this spatial information in the parietal cortex, it's then transferred to the premotor cortex, where it's used for action planning and actual movements. So this picture is showing us a very thin slice of the brain, what's called a sagittal section, and cell bodies are stained purple. And so each little purple dot here is one cell, one neuron. We can see that there are different parts of the brain that have different densities of cells. The darker areas are areas of very densely packed cells, while some areas are less densely populated. So let's zoom into the cellular level and see how single unit recording works. So this is how we find information that neurons carry. This is a little diagram representing electrodes that are very small. They're wound together and carefully lowered into the brain area of interest. They're about the size of a cell body, and within a small range, they're able to sense electrical potentials. And if they're close enough to cells, that is, if we put them in a densely packed area, then we're able to record the action potentials of single neurons. And an action potential is an electrical impulse that moves from the cell body to the cell's axon and allows communication between cells, allows information to be transmitted from one cell to its neighbors. And so by looking at how many and what rate at which neurons transmit these action potentials, or I'm going to say fire action potentials, then we are able to find out what type of information each single neuron in the brain is able to carry. So, let's zoom out a little bit to see how this works at the population level. So, in the early 70s, neurophysiologists were able to record from cells in the hippocampus, and what they were able to demonstrate is that this part of the brain represents an abstract reference frame of space. So, this diagram here is recordings from 18 cells in the hippocampus, and each square represents the entire space of the observable environment. So that's like the room that we're in, or the room that the animal is in when it's being recorded from. So each of these blue areas is a space where the cell has a very low firing rate, and the hotter areas, the red areas, are where these cells are firing many action potentials. So we can see that in conjunction, a large number of these cells are able to map out the space of the entire observable environment. So what these cells do is when an animal is in the location of their firing field, they'll send many action potentials, and what they do is they track where the animal is in the environment, irrespective of the behavior. So these cells are just tracking where the animal is, and we call these place cells. So it was not explained how these cells operate or how they create this allocentric reference frame until about 2006, when there was a discovery of grid cells. So grid cells also map an allocentric reference frame of space, just like the hippocampal place cells. However, they're a little bit different. Each one of these diagrams also represents the entire space of an environment, the room, from a bird's eye view. But you can see that this represents 5 different cells, and each grid cell has its own distinct grid pattern. So they have different levels of maybe zoom, you might say, or so they each represent a constant grid pattern in the environment, but each one is distinct. And some of these cells are actually sensitive to the head direction of the animal, or what direction the animal is traveling. And so, like this cell in the middle here, it would only fire in this grid pattern when the animal is moving through those points on the grid in a particular direction, represented here by these arrows. So this is in the entorhinal cortex. We were in the hippocampus. This is found in the entorhinal cortex, grid cells. So, when we superimpose all the firing fields of these 5 neurons, we get this map here. And through the application of a threshold, we can see that place fields are created. And this happens in the interaction between the entorhinal cortex and the hippocampus. Yep. So there are 2 other important experimental findings that tell us how these place fields work. So the first is, if an animal is placed in an environment and all the lights are turned off, then we still see the grid pattern in the entorhinal cortex. However, over time, as the animal is moving around, these grids become less well defined. So we know that visual information is important, but the vestibular information, that's the head direction, is actually more important for the creation of these grids. The second experimental finding that's very important is that when the connection between the entorhinal cortex and the hippocampus is broken through a lesion, we do not see the firing fields, these place cell firing fields. So we know that both entorhinal cortex and hippocampal allocentric reference frames depend on the self-motion information of the animal. Now we're going to move into the parietal cortex now. So in the parietal cortex in 2006, it was discovered that cells actually map another allocentric reference frame that's distinct. Now this reference frame is in relationship to the space of a route. So if we put an animal, if an animal is trained to run on this type of maze here, it's trained to run back and forth, and we can linearize this maze and plot out the firing rate of a single cell in the parietal cortex. And what we find is that although the behavior is changed when the animal is running outbound versus inbound runs, we see the same firing rate, we say the same tracking of space of the route. So it doesn't matter where these mazes are placed in the environment. We see a constant firing pattern for the parietal cells along the route. So my question is, how does the parietal cortex create these route maps? And there are two hypotheses that come from our diagram here. So looking at the inputs to the parietal cortex, we see that there's some input from the thalamus, that's sensory information. And we see that there's also retrosplenial cortex input that comes from the hippocampus. So the two hypotheses are, number one, maybe the parietal cortex is taking this spatial information from the hippocampus that's irrespective of behavior, and using that information, integrating it over time to create a strictly spatial map of the route. Also, it could be that other inputs, such as the input of the sensory information from the thalamus, is also being integrated with this spatial information in order to create the route map. So, in order to answer these two questions, I built what's called a generalized linear model to analyze some specific data that's already been collected by Dr. Nitz in this lab. And before I tell you about how the model works, I'm going to tell you about what the experimental setup, what type of data we're analyzing. So first of all, animals were trained to run on two different mazes. And here we have actual position points of animals at a constant time frame while they run on our two different mazes. One is shaped like a top hat, and one is shaped like a W. So they have a little bit different sequences of actions that the animal has to go through. So in order to build the generalized linear model, my first task was to take this raw position data that's time-stamped and extrapolate measures of self-motion like velocity and head direction. And I can sum that up in one sentence, but that was a pretty tough task and it took a lot of work. So once we have these measures of self-motion, I also created some predictor factors that represent the space of the route. So what I did to build this generalized linear model, so what a generalized linear model does is it takes in multiple predictor factors and tries to create a model of what is in this case the firing rate of a cell. So it considers, in my case, we have 8 predictor factors, some representing the space of the route, some representing the self-motion information. And it takes these predictor factors, and what it does is it multiplies a constant coefficient by each one of these predictor factors in order that when all the predictor factors multiplied by their coefficients are added up, we have a model that's as close to the firing rate of the cell as possible. And then what we do is we measure how close that model fits to the firing rate of the cell, and then we take out each predictor factor individually, so that we know that if we take out the velocity factor and the model degrades a lot, then we know that the velocity factor is an important factor for the firing rate of the cell. And so the output of the generalized linear model looks something like this. So in blue here we have the firing rate of the cell, and in red we have the model that I created with all 8 predictor factors. Oh, sorry, I didn't mention what predictor factors are we're looking at. So we have the velocity, the acceleration, the head direction, and the angular velocity. We're calling these self-motion predictor factors. And then we also have spatial factors. That's the route position. The route position is just the numbers 1 through 206. And we also have the probability of being at different types of turns. So probability of being at a left turn, probability of being at a right turn, or the probability of being at the center of a straight. Okay, so 8 predictor factors. Some represent self-motion, some represent space. And what happens is we create a model. The model looks somewhat like the firing rate. And then, of course, we take out each individual predictor factor. And so here we have the red is the total model with every predictor factor. And we see that when we take out one predictor factor, in this case, the velocity of the animal, we see that the model degrades a little bit. It's different. It does not do as great of a job of representing the firing rate of the cell. So then we measure the correlation between each model and the firing rate of the cell, and we see the difference or how much the model degrades when we take out each predictor factor. So for each cell, this is one cell, for each predictor factor, we have a graph that shows the relative contribution of each predictor factor to the firing rate of each cell. So we have a couple hundred cells, so we have a lot of graphs that look like this, and we can look on an individual cell basis at what predictor factors are important for the firing rates. Now, in order to look at a large group of cells, in order to kind of find out what the parietal cortex as a whole is doing, what we do is we test for significance for each predictor factor. So this graph, what we have here on the y-axis is each cell. So each row of this graph represents a single cell, and this is just a test for significance. This is not the relative contribution. This is if the contribution of each predictor factor is important. That's statistically significant for the firing rate of the cell. So on the bottom here we have each of our predictor factors. Self-motion are the first four, and 6 through 9 are the position factors, the spatial factors. And what I've highlighted here is we also looked at all the self-motion variables. So we created another model that looks at when we take out each of these self-motion variables, how many cells have a significant portion of their firing pattern described by all the self-motion variables. And this last column is how many cells are whose firing rates are significantly important based on the spatial variables. Okay, so what this graph is telling us, right, is that for a lot of the cells, both self-motion and spatial variables are important for the firing rates. That's statistically significant. So what's this mean for our initial question, which was how does the parietal cortex create route maps? That last graph was showing us that both the spatial information and the self-motion information were important for the firing rates of most cells. So I'd just like to thank my advisors.
Awesome.
Any questions? Yeah.
I guess I was wondering about, so when you did the analysis of the ability of your model to predict data, I'm assuming, did you do some sort of cross-validation where you use some of the data to train the model and other cells to test the model?
Okay, so this is it's a mathematical model to describe to try to describe the firing rate. And no, we didn't. There was no like testing phase and trial phase and testing phase. It was just we put all these predictor factors in, and we got a model, and we analyzed how good that was based on correlation between the firing rates and the model.
Do you think it would be interesting sometime to combine the lesion approach with of different locations that are feeding in, and to see if, that you talked about, and to see if that actually, you could model that as well, to predict what then the contribution of each of those.
Right, yeah, so the idea is that the hippocampus, because the firing rates of the place cells are irrespective of the behavior of the animal, we just kind of assume that the spatial information from the hippocampus is not carrying self-motion information. But a lesion study, if we cut that connection between the retrosplenial cortex and the parietal cortex, could be able to show whether just the thalamic input is important, or whether the parietal cortex receives some self-motion information from the hippocampus, which is definitely a possibility. Yeah. So it would be very interesting to do a lesion study. Yeah.
Thank you.